Looking at the second step of the central dogma of biology, it is translation. Now, translation involves the conversion of an RNA molecule into protein. There's 3 steps in translation. The first step being initiation, second step elongation, third step being termination. There are 3 key RNA players in translation. You have mRNA, tRNA, and rRNA. Now, we already know mRNA and what that does. Looking at tRNA are molecules that help bridge the gap between the codons of the mRNA and the amino acids they encode, whereas rRNA helps to catalyze the formation, or helps to catalyze the bonds between the incoming amino acids that gives rise to the polypeptide chain. Now, looking at the ribosome itself, it's made up of 2 subunits. You have the large and the small. Within the large subunit, you have the E site, the P site, and the A site. Now, looking at the first step of translation, of initiation, this basically involves the formation of this ribosome complex. Now, what happens is you have your mRNA, once it leaves the nucleus and it's just been transcribed, it finds its way into the cytoplasm. And when it's in the cytoplasm, the small ribosomal subunit finds itself to the beginning strand or the beginning sequence of this mRNA strand, and this initiates the large ribosomal subunit to come in and form this complex. Now, looking at these E, P, and A sites here, this is going to involve the elongation step of translation, and that basically involves the formation of that polypeptide chain. Now, incoming tRNA molecules, they're known to be charged whenever they contain their amino acid. Once the charged tRNA molecule enters the A site, the ribosome catalyzes the transfer of the amino acid that is attached to the tRNA molecule in the P site to the amino acid attached to the tRNA molecule in the A site. Now, once this transfer occurs, the tRNA molecule in the P site transitions to the E site, while simultaneously the tRNA molecule that was in the A site transitions to the P site. Now, this elongation process continues to occur until a stop codon is reached in the mRNA sequence, and once that stop codon is reached, a release factor comes in and binds to it, which causes the ribosome complex to fall apart and the polypeptide chain to be released.